I'm David Hagele from University of Stuttgart, and I'm presenting this joint work that was done in collaboration with Yushen Tang and my supervisor Daniel Weiskopf. Um, yeah, so thanks Stefan for this introduction, so I don't have to do a lot of this again. So for the BioMedVis challenge, this visualization was given basically. So it's a scatter pie chart, and it represents the cell type mixtures at different locations on cellular tissue. So each pie chart represents a mixture of different cell types, and this whole visualization leverages human perception for pattern recognition. And for example, by looking at the visualization, we can observe regions on the tissue that exhibit similar mixtures. And the scatter pie visualization is superimposed onto a histological image so that analysts can relate the observed patterns to the tissue. So now, while this whole visualization is effective, it comes with a few drawbacks. For example, the histological image is occluded by the pie charts. Also, the perception of the pie chart colors is influenced by the background image, so that the patterns of the tissue structure interfere with the patterns of the scatter pie chart. Comparing different mixtures is difficult, which is a general drawbacks of pie charts. And finally, the similar mixtures can only be easily found locally, but it's very hard to spot similar mixtures that are spatially further away from each other. So this brings us to the redesign of this visualization. As a first step, we choose to separate the histological image from the cell type mixtures. And next, we want to replace the pie charts with a different representation of the mixtures. So for this, we choose stacked bars, which also provide a nice parts of a whole representation. And these bars are then displayed next to each other, as in a regular stacked bar chart. Since we lose the spatial information now by doing so, we provide linkage between the views so that the mixtures can still be related to the spatial context of the tissue. So the user is able to select a bar and get a highlighting on the histological image, or the other way around. And selecting parts of the image highlights the corresponding mixtures in the bar chart. So with the current redesign, we also have thrown away the human pattern recognition part that was possible with these scatter pie charts. So we need now a replacement that allows to find similarities among the mixtures. And our approach is to move this responsibility to the computer so we can apply our trusty pattern recognition algorithms. However, first we need to give a little thought on how to measure the similarity between cell type mixtures. So what is the correct metric to use? And we were inspired through other research that deals with the analysis of such kind of data, and decided to use the Aitchison distance. So what is the Aitchison distance? This is a metric introduced by John Aitchison and is intended for compositional data. Compositional data is living in a special space where the parts of a whole sum up to 1. And this is different from standard Cartesian coordinates, since when one component of a vector changes, one or more of the other components also have to change in order to satisfy their sum resulting into 1. And for this special geometry, the Aitchison distance provides a metric that compares the components' ratios instead of their values. So now we are equipped with some tools for compositional data, and will apply these to the data. So first we transform the data into a space with regular arithmetic, where the regular arithmetic operations preserve the compositional nature of the data, and where the Euclidean distances correspond to the Aitchison distance. So this is done with the center log ratio transform. And then we can apply classical pattern recognition algorithms such as k-means, which provides a clustering of the cell type mixtures, And we also apply dimensionality reduction of the cell type mixtures, which gives us a third view where the mixtures are arranged according to their similarities. So for this we use PCA, just standard PCA in the center log ratio coordinates, which works pretty well and shows many different blobs. So as can be seen here in this right upper plot. So in the end, we have built this visual analytic system with brushable linked views. We have the histological image on the left here, and the cell type mixtures on the bottom. These are like the stacked bars. And the dimensionality reduction view here. The clustering from k-means is used to color the dimensionality reduction plot. So here we have 3 clusters currently. You can ramp this up basically. So with blue and orange and yellow cluster here. And since this system heavily relies on the brushing and linking for exploration, I also brought a video showcasing the interaction. So let's see. All right. So we have the 3 views, and we can show and hide the spot markers here. We can ramp up the number of clusters for k-means, where we can see that the clusters belong to coherent areas on the tissue. We can now select all the mixtures from a specific cluster. On the bottom of the bar chart has updated for the selection there. We can also do selections in the dimensionality reduction plot, see the corresponding tissue parts and corresponding pattern of the mixtures basically. Yeah. You can also select parts of the tissue and see what kind of mixtures are in there. Yeah, of course, selections in the bars are also possible. Yeah. So pretty much this, just the power of linked views basically applied to this data set, I think. And of course we have like this, a table view on the bottom where you can like sort and filter for stuff that like for the numerics that you like. All right. And with this, I'd like to end my talk and thank you for the attention. Yeah, that's it.